All right. Good morning, everyone, and thank you very much for joining today's webinar entitled Large-Scale Genome Editing for Metabolic Engineering of E. coli. My name is Laura, and I'm the Marketing Specialist for the CRISPR services here at GenScript. I'm pleased to introduce our presenter today, Dr. Yifan Li, who is our Senior Scientist with years of metabolic engineering experience. Microbes, such as E. coli, have many applications in biotechnology, including protein, chemical, and biofuel production. And as gene editing technologies continue to advance, the potential for these microbes to accelerate both research and industry will also increase. And in this webinar, we'll describe the methods and practical applications for metabolic engineering in E. coli. So if you have any questions during the presentation, you can submit them by typing them into the questions field that you see on your screen. And we'll do our best to answer them after the webinar. Frequently asked questions, as well as their answers, and the webinar itself, will be posted later on our website at www.genscript.com. And then finally, at the end of the presentation, you'll receive a brief survey. We encourage you to fill this out because it'll help us better design and optimize our services to meet your needs in the future. And so with that said, I will now turn the presentation over to Yifan.
Okay, thank you, Laura. As Laura mentioned, the topic of today's webinar is large-scale genome editing for metabolic engineering of E. coli. My name is Yifan Li, and I'm the Senior Scientist of GenScript. To begin with, I wanted to quickly go over the content of today's presentation. To start, I will give a brief introduction of metabolic engineering and some of its applications. Next, I will discuss some of the commonly used methods for genome engineering of E. coli. After this, I will describe our CRISPR technology for genome editing of E. coli, its technological basis and advantages. At last, I will give some case studies on how to use our CRISPR technology for metabolic engineering applications. So, what is metabolic engineering? Metabolic engineering is a process of altering the expression of genes or adjusting regulatory pathways in the cells to increase the production of specific compounds. Microbes are ideal candidates for this, since they are relatively easy to culture and manipulate, and therefore has the potential to significantly decrease the cost to synthesize some of the value-added substances. There are a variety of substances that have been produced using metabolic engineering, including chemicals, drugs, and fuels. For instance, 1,3-propanediol has been commercially produced by the DuPont company using recombinant E. coli. Amino acids have also been produced at multi-million-ton scale with engineered microbes. In addition, the Amyris technology has engineered Baker's yeast strain to produce never-before-seen concentrations of artemisinin, which can be used for the cure of malaria. Finally, the production of bio-based ethanol, ethanol, and fatty acids are key for the sustainable supply of alternatives to petroleum-based fuels. Despite the potential, microbiome has unfortunately not evolved to suit the practical outcomes desired by the humankind. Thus, when microbes are isolated from nature, their efficiency in producing any given molecule is rather low. So, we need to engineer the cellular metabolism to improve the productivity. In this proof of concept illustration, if we want to improve the production of the desired products, we need to introduce multiple genomic modifications. For example, we may need to block the competing pathway, here designated as M, to increase the flux to the product. We might need to overexpress the related metabolic pathway genes from A to B to C. If one or several enzymes in the pathway are missing in the cell factory, we might have to introduce these genes into the cells to complete the pathway, here designated as the pathway from D to the product. if negative regulation mechanisms exist in the cell, for example, the accumulation of a certain compound may inhibit the expression of key enzymes. In this case, we have to remove this negative regulation for efficient production. To accomplish any of these modifications, genome engineering technologies are essential. Let's consider a real case study. DuPont Company has engineered E. coli strain to produce 1,3-propanediol, also known as bio-PDO, which has been a benchmark story in metabolic engineering. For developing such an efficient cell factory, scientists in DuPont overall introduced 26 modifications in the genome of E. coli to modify the metabolisms of a variety of pathways. This cannot be accomplished without efficient genome engineering technology. Next, let's move on how genome editing is accomplished in E. coli. Recombination-mediated genetic engineering, also known as Recombineering, has until recently been the most commonly used system for genome engineering of E. coli. Recombineering uses the bacteriophage lambda-red system to help with homologous recombination. The lambda-red system includes 3 phage recombination genes: gamma, beta, and exo. Exo has 5:3 double-strand DNA exonuclease activity, which can generate 3 overhangs on the liner DNA. Beta-protein binds the single-stranded DNA, promoting single-strand annealing and the generation of a recombinant DNA. An additional protein, gamma, prevents the REC-BCD nuclease from degrading introduced liner DNA fragments. Working together, these 3 proteins insert liner DNA at the desired target, creating genetic recombinants. Based on the Recombineering system, several genomic modification methods have been developed, including the method based on PCR targeting, positive-negative selection, FLP, FRT site-specific recombination system, the SCE1 endonuclease system, the MAGE technology developed by George Church, and the most recently developed red CRISPR technology. On this slide, I've outlined these major genome editing methods, along with some of their advantages and disadvantages. For instance, PCR targeting, positive-negative selection methods, and FLP, FRT are great for gene knockout. However, they have limitations for gene knock-in. Also, with the exception of positive-negative selection, these techniques have to leave behind a scar which can potentially compromise the genetic background of future modifications. While SCE1 and MAGE have improvements over these techniques, the lambda-red CRISPR system is still the most ideal technology. In this system, the marker is reusable, it's scarless, gene knockout or knock-in is not limited by gene size, multi-gene targeting is possible, and it has the fastest turnaround time. On the next few slides, I will spend a bit more time describing some of these traditional recombination methods, as well as the red CRISPR technology. The most commonly used recombination method is the FLP, FRT-based method. In this system, a selectable marker is used to replace the knockout gene through a double crossover event. A selectable marker is required because the recombination frequency is very low, so a marker will be needed to select the recombinants. A selectable marker is flanked by 2 FRT sites. In the second step, the expression of FLP recombinase stimulate the recombination between the 2 FRT sites, removing the selectable marker from the chromosome. This method allows repeated genomic modification using the same marker, but it has 2 disadvantages. One disadvantage is that it requires 2 recombination steps for markerless gene knockout. The second is that an FRT site has to be left in the targeting region. It is like a scar and is undesired in some real-case applications. Some applications require precise and seamless modification of a target locus, so the FLP, FRT method is not suitable for this. One strategy to solve this problem is to add a counter-selectable marker to the donor DNA. Generally, a counter-selectable cassette along with the selectable marker, is first inserted at the target site. In this case, the selectable marker is a cat gene, and the counter-selectable marker is the STAC-B gene. Then, this cassette is replaced with the desired alternation in the second recombination reaction. When cultivated on sucrose, the expression of STAC-B gene is lethal to E. coli, so placing the cells on sucrose helps to select the recombinant strains. This two-step selection counter-selection procedure allows insertion of the desired DNA without leaving any undesired modifications in a seamless manner. But this procedure is very time and labor-consuming. Another strategy to seamlessly introduce genomic modifications is taking advantage of the SCE1 site-specific endonuclease. This endonuclease recognizes and cuts at a specific sequence. In this method, the selectable marker is flanked by two SCE1 cutting sites. The cutting of the genomic sequence introduces double-strand breaks in the genome, which can stimulate recombination and select marker-evicted strains. However, this method requires to pre-introduce the SCE1 recognition site into the genome, so it requires at least 2 steps of recombination. The bacterial CRISPR-Cas9 genome editing technology is a revolutionary technology and has been used in diverse organisms, including bacteria, yeast, and human cell lines. This system is able to introduce double-strand breaks at any desired locale in the chromosome. The functionality of the system relies on the proper expression of the Cas9 protein and a guide RNA. Another important factor for successful binding of the Cas9 is a PAM sequence, which locates immediately following the target sequence and has a canonical NGG pattern. The binding of the gRNA Cas9 complex localizes Cas9 to the genomic target sequence, so the wild-type Cas9 can cut both strands of the DNA, causing a double-strand break. In mammalian cells, the double-strand break can be repaired through 2 general repair pathways. The first is a non-homologous end-joining DNA repair pathway. The second is a homology-directed repair pathway. But in most prokaryotic cells, the non-homologous end-joining is very inefficient, so the double-strand break has to be restored by a recombination event. So, to increase CRISPR editing efficiency, the CRISPR system can be combined with lambda-ready Recombineering system to ensure the double-strand break repair in E. coli. Unlike previously described methods, the red CRISPR system, which is used as GenScript, requires only a single recombination step to introduce the desired modification. No selectable marker is needed to integrate it into the chromosome to select the recombinant. To achieve this, CRISPR system introduces a double-strand break at the targeting site. This double-strand break has 2 functions. First, it helps to introduce the recombination efficiency. Second, it kills non-edited cells to leave only the recombinant cells, so it actually serves to select the recombinants, allowing the circumvent to use a selectable marker. Using this method, we routinely get more than 90% efficiency for ordinary genome editing. Another key advantage, as I mentioned earlier, is that you can edit multiple genes simultaneously. So, in the next section, I will describe how CRISPR-mediated genome editing can benefit your metabolic engineering applications. To illustrate this, I wanted to present a real case study. This work describes genome engineering of E. coli for the production of beta-carotene. Beta-carotene is a red-orange pigment, belongs to the isoprenols family, and has many applications in pharmaceutical industry. For instance, beta-carotene has been investigated as a possible cancer therapy. In this example, E. coli is engineered to synthesize beta-carotene from sugar. 2 important pathways work together to synthesize beta-carotene from intermediate metabolites in the cells, the MEP pathway and the beta-carotene biosynthetic pathway. Sugars, which are broken down during the glycolysis to form glucose 3-phosphate and pyruvate, are converted to FPP through the MEP pathway. FPP is then converted to beta-carotene through the beta-carotene pathway. The MEP pathway shown here is a 9-step pathway. The genes catalyzing the steps are native genes in E. coli. On the contrary, the CRTEBIY genes catalyzing the beta-carotene biosynthetic pathway are absent from the native chromosome. So, to improve beta-carotene production, we have to take 2 steps. The first step is to introduce the CRTEBIY genes in the host, which will facilitate the synthesis of beta-carotene from FPP. The second step is to optimize the expression of key genes in the MEP pathway to increase the flux to beta-carotene production. In the first part of this process, 4 genes, CRTEBIY, are integrated into the chromosome at the LDHA locus using red CRISPR. CRISPR generates a double-strand break, facilitating the knocking in of the whole pathway. As mentioned previously, there are 2 key advantages to this method. First, gene knock-in can be accomplished by CRISPR in a single-step recombination, and also no antibiotic selection is required. E. coli strain with this integrated pathway were able to produce the yellow pigment beta-carotene. An alternative strategy, and also a commonly used strategy is to use a plasmid to express the CRTEBIY genes. But a straightforward disadvantage of plasmid-based expression is that the expression is unable and requires a selectable marker to maintain the plasmid. Antibiotic selection will also take a great amount of effort and cost in industrial applications. After integrating the CRTEBIY genes into the genome, the next step is to increase beta-carotene production. The next step is to increase beta-carotene production by increasing the flux to the MEP pathway. In other words, that is to direct more carbon resources into the MEP pathway. To do this, an obvious strategy is to overexpress the genes in the MEP pathway. This has been done previously by another group using traditional recombination system. They have used the strong T5 promoter to replace the native promoter of these genes. Using this approach, there were a few key disadvantages. First, a marker has to be introduced into the recombinant strain, which in turn requires an additional recombination step to remove the marker. The whole process for generating a single modification takes about 2 weeks. In addition, an FRT site will be left behind in the promoter region, interfering gene expression and subsequent genomic modifications. Using red CRISPR, the genome-based gene overexpression can be accomplished much easier. In this system, the author used the red CRISPR to cut the native RBS region and use a strong promoter and a strong RBS to replace the region. This promoter replacement can also be achieved in a single recombination step. The author overexpressed all pathway genes in a sequential and combinatorial manner, and collectively constructed 33 different strains. This combinatorial search yielded a strain producing 80 microgram per liter beta-carotene, which is 3-fold higher than the starting strain. In addition, a strain with all 9 MEP pathway genes overexpressed were constructed. Normally, if you use traditional recombination method, it could take up to half a year to introduce these 9 modifications. But using red CRISPR, this work can be completed in a month. Up to now, we have described the methods for gene deletion and overexpression, but there might be some cases in which we don't know the best expression level of a certain gene. For example, an enzyme may catalyze a branch point reaction. If the expression level of the gene is too weak, it may not direct enough carbon resource to the desired products. On the other hand, if the expression level is too strong, the resources needed for cell growth will be insufficient. Again, not good for the production. Only when the expression level of the gene is in a certain medium level, the cell factory will reach the best performance. So the goal is to tune the gene expression level to find the best solution. To accomplish this, I would like to introduce a powerful software system we have in-licensed from Dr. Howard Salas at Penn State University. That is the RBS calculator. This software is able to design the RBS sequence with a specified strength. As a result, we can use a collection of RBS sequences spanning a wide range of different strengths. If we use this collection of RBS sequences to replace native RBS, we get a collection of strains, and in each of the strains, the targeted gene is expressed at a different level. So by screening this collection of strains, we can get the best strain with the most suitable gene expression level. In addition, with our red CRISPR technology, the promoter replacement can be achieved in a single-step recombination. In summary, our red CRISPR technology can provide 4 different solutions to your metabolic engineering applications. You can use red CRISPR technology for gene knockout, for gene overexpression, for gene knock-in or pathway knock-in, and for fine-tuning gene expression level. Essentially, this technology can meet almost all genome editing requirements in the optimization of your cell factories. Finally, I would like to briefly introduce another service that could accelerate your metabolic engineering projects, our metabolic pathway assembly service. This service is based on our in-licensed OMA technology, and with this technology, we can provide both rational pathway assembly and the pathway library assembly service. For pathway library assembly, we can use a collection of RBS sequences to fine-tune several pathway genes simultaneously. If you'd like more information on this service, please visit our website I have listed on this page. So now I will turn this presentation over to our marketing specialist, Laura, who will describe a few of our microbiome genome editing service features. Thank you.
Great. Thank you very much, Yifan. All right, thanks again, everyone. So to wrap up this presentation, I just want to take a few minutes to talk about some of the services that might help support you in your metabolic engineering projects. Okay, so here at GenScript, we offer 2 services that feature the lambda red CRISPR recombination technology that we talked about. We offer both a knock-in and a knockout service for E. coli strains. For both services, you will provide us with the starting strain name and the target gene name and sequences. We will then provide you with your recombinant strain and glycerol stock, along with detailed quality control reports. The turnaround time is one of the fastest in the industry, starting from just 4 weeks, and pricing starts from $4,000 per strain. And if you'd like any more information about these services and to learn about any updates that might be coming, you can visit the address that I have listed here. So in terms of workflow, there are 4 main stages for the generation of our knockout and knock-in recombinants. In the event that a knock-in is requested, we can synthesize your target gene at an additional cost. We have a license with the Broad Institute, and as part of the license, have access to proprietary gRNA design tools that minimize off-targeting. We will design and construct the gRNA for you, carry out the CRISPR gene editing, and then finally validate your strain using PCR screening and sequencing. And I also wanted to share a few more resources at GenScript that you might find useful. GenScript offers a wide range of CRISPR products and services, from CRISPR plasmids to gRNA libraries and mammalian cell lines. We also provide many free technical resources, including webinars, which I'll mention in a sec, and protocols that you might also find handy. Finally, we just finished up a new CRISPR handbook, And this is available as a free PDF and provides both background on CRISPR as well as helpful tips for applying it to your research. So please visit our website that I've listed here, www.genscript.com, CRISPR, for access to all of these resources. So I hope that you found this webinar interesting and useful. And before we start taking questions, I just wanted to let you know about some other CRISPR webinars you might want to check out as well. If you're interested, we have another webinar coming up on the applications of CRISPR-Cas9 for disease research, presented by Randall Platt and Fang Zhang's lab. You can register for this free webinar by visiting our website, GenScript.com, and clicking on the webinar link on the home page. On our CRISPR resource page that I also have here indicated at the bottom of the slide, we have access to other webinars that we've previously aired. You can either watch them or you can download the PDFs for free. So at this point, I'd just like to thank you guys again for attending. We hope this is useful. At this point, we would be happy to take any questions that you might have. Again, if you want to ask them, you can just enter them into the questions field, and we will do our best to answer them. Okay, so the first question is, why does CRISPR need to be combined with lambda-red recombinaring? Wouldn't it be suitable for gene editing just by itself? Yifan, do you have any comments?
Okay. For mammalian cell line engineering, CRISPR in itself is sufficient to introduce the gene knockout, and the double-strand break can be repaired by the non-homologous enjoining pathway. But in E. coli, the non-homologous enjoining mechanism is inefficient, so CRISPR in itself is not sufficient for modification. The lambda-red system have to be utilized to facilitate the recombination. That is why lambda-red have to be used.
All right, great, thanks. Okay, so we have another question. This time it's about the OLMA technology that we talked about. So the question is, how does the OLMA technology work? How is it different from other assembly methods like Gibson assembly or Golden Gate assembly?
It is a very complicated question. OLMA technology is actually based on Golden Gate assembly. The mechanism is very similar. The difference is that OLMA technology use we call it linkers, use linkers to help the assembly, and some regulatory sequences such as RBS and promoter can be added in the linkers. So if we want to fine-tune the expression level of the pathway genes, we can use different RBS sequences to control the expression of a certain gene. So OLMA technology is especially powerful for the construction of pathway libraries and to tuning the gene expression levels in the pathway.
Great, thank you, Yifan.
Thank you.
Okay, so we have, looks like we have another question. This is actually now about the RBS calculator. So how does the RBS calculator work? Why would you need to use an RBS calculator instead of just using a standard set of promoters of different strengths?
RBS, Dr. Howard Salas developed the RBS calculator based on the, I guess, the biophysical models of the interaction between the ribosome binding site with the ribosome. I think the mechanism is complex. And why do we use RBS calculator to design RBS sequence for different genes rather than use a single set of RBS collection? This is a very good question. As far as I know, the same RBS, the strength of a particular RBS sequence can be different when it initiates the translation of different genes. In other words, if you place a specific RBS sequence in front of different genes, the strength can be totally different. So we have to design an RBS sequence for different protein coding sequences.
Okay, great. Thanks, Yifan.
Thank you.
So I guess a follow-up question to this would be, do we help to design the RBS sequence if we want to use the library to tune the expression levels?
Yes, we can help to design the RBS sequences, and it is free if you order the genome editing service from us.
Okay. Thanks, Yifan. Okay, so that looks like that's the rest of the questions that we've received. So I think at this point what we'll do is wrap up the webinar. If you have any more questions, feel free to ask them now. We have a little more time. Otherwise, if you have more that you'd like to maybe ask later, there are ways that you can still ask them. You can either email either Yifan or myself at the email addresses I have listed here. Or we have a lot of technical resources regarding the topics that we talked about today. So you can visit either of the two web pages that I've listed here to get some more information. So with that, I would, sorry, one more question. Can we get the presentation slides? Yes, you can. We'll be sending out an email to everybody with a link to the webinar, as well as you can download the PDF as well. So we'll send you guys a link, and then it'll also be posted on our website very shortly. So if you missed any of the slides that we presented today, we will have both the recording and the PDF available for download. So good question. All right, so with that, thank you again very much. We hope this was useful to you, and good luck with your experiments.